Hi everybody. Now I discuss about sterilization. First of all, introduction. Sterilization is defined as the process by which an article, surface or medium is free from all microorganisms, either in vegetative cells or spore state. Aims of sterilization: to prevent microbial contamination, to maintain aseptic conditions, to ensure safety from contaminating microorganisms. There are two types of sterilization. One is physical agent, another one is chemical agent. Physical agent: heat, dry heat and moist heat, radiation, filtration. And chemical agent: alcohol, halogens, phenol and gases. First of all, physical agent. Dry heat. Dry heat is not as effective in killing microorganisms than moist heat. Microorganisms are killed by destructive oxidations of essential cell constituents, protein denaturation, and toxic effects of electrolytes. Killing the moist resistant spores by dry heat require a temperature of 160 degrees Celsius for 2 hours. Types of dry heat methods: flaming, incineration, hot or oven. Flaming with red dot, without red dot. With red dot, inoculating loops, wire, forceps, spatulas are held in Bunsen flame till they become red hot in order to be sterilized. Then without a red dot, scalpels, needles, mouth of culture tubes, glass slides, cover slips, etc. could be passed a few times through the Bunsen flame without allowing them to become red dot. Incineration. Objects are burned in direct flame, suitable only for disposable items. Hot or oven. The most widely used method of sterilization by dry heat, hot or oven, is a double walled metal chamber which is electrically heated. A holding period of 160 degrees Celsius for 1 hour. It is used to sterilize glasswares, forceps, scissors, scalpels, all glass syringes. It is not suitable for fabric materials of dry heat, particularly in hot or oven. Then moist heat. Moist heat is much more effective than dry heat because it kills microorganisms by coagulating and denaturing their enzymes and structural proteins. Sterilization requires exposure to moist heat at 121 degrees Celsius for 15 minutes. The major principle involved in moist heat is steam under pressure. There are types of moist heat. Moist heat at below 100 degrees Celsius, moist heat at 100 degrees Celsius, moist heat below 100 degrees Celsius for boiling. Water bath used for sterilizing material which cannot be heated at 100 degrees Celsius or more. Example: serum, body fluid, or vaccines. Tindalization. It involved heating to 100 degrees Celsius and return to room temperature for 3 successive days, useful for killing spore formers. Pasteurization. This process involves controlled heat treatment. It can be carried out in two ways: by heating at 63 degrees Celsius for 10 minutes is the low temperature long time, and high temperature short time for 72 degrees Celsius for 20 seconds. This is suitable only juice and milk. Moist heat at 100 degrees Celsius. Boiling the materials in water for 10 to 15 minutes. Kill all non-sporing organisms. This method is used to sterilize forceps, scissors, syringes, needles, gloves, etc. The Koch or Arnold steamer is used for moist heat at 100 degrees Celsius. This method is inexpensive method and steam under pressure. This is the principle of autoclave or steam sterilizer is that water boils when it vapor pressure equal that of the surrounding atmosphere. Sterilization by steam under pressure is carried out at a temperature between 108 degrees Celsius and 147 degrees Celsius. Several types of steam sterilizers are used for laboratory autoclaves, hospital dressing sterilizer, bowl and instrument sterilizer, rapid cooling sterilizer. Then radiation. There are two types of radiation. One is ionizing radiation, another one is non-ionizing radiation. Non-ionizing radiations involved for infrared rays and UV rays. They have longer wavelength and produce high heat. Infrared rays are considered as hot air sterilization. Infrared rays are used for rapid mass sterilization of syringes. UV rays is used for disinfecting enclosed areas, entryways, hospital wards, operation theaters, and laboratories, laminar airflow chamber. Ionizing radiations, these rays are lethal to DNA. They have very high penetrating power. It is used as cold sterilization because it produces low heat. It is used for sterilizing plastics and syringes, swabs, culture plates, catheters, etc. Example of ionizing radiation: X-rays, gamma rays, and cosmic rays. Filtration. This is a useful method for sterilizing antibiotic solutions, sera, and carbohydrate solutions, vaccine toxins in the preparation of culture media. This method, microorganisms are filtered in the filter disc. Then chemical agent. Disinfectants. Several chemical agents are used as antiseptic and disinfectants. This mechanism involved for this agent a wide spectrum of activity and be effective against all microbes. Be effective in acid as well as alkaline media. Have high penetrating power. Be stable, not corrode metals. Be expensive, easily available. Be safe and easy to use. First of all, alcohol. Ethyl alcohol and isopropyl alcohol are the most frequently used. They are mainly used as skin antiseptics. They denature the bacterial protein. They have no action on spores. To be effective, they must be used at a concentration of 60 to 70 percentage in water. Isopropyl alcohol is used for disinfections of clinical thermometer. Methyl alcohol is effective against fungal spores and it is used to treating incubators and cabinet. Next is allergens. Iodine and chlorine are allergens. Iodine in aqueous and alcoholic solutions have been used as skin disinfectant. It is active against tubercle bacilli and viruses. Chlorine is used as a disinfectant in water supplies, swimming pools, food and dairy industry for many years. Hypochlorite is a bactericidal effect and have wide spectrum of action against of viruses. Phenols. It is the first widely used antiseptics. Is the antiseptic surgery the father of Joseph List. Phenol components, creosols and xylenols are used as disinfectants in hospitals and labs. Commercial disinfectants lysols is the mixture of phenolic compounds. Phenol will denature the protein and disturb the cell membrane. Chlorohexidine is a non-toxic skin antiseptic. Phenols are effective in the presence of organic matter and remain active for a long time. Phenols can cause skin irritation and have a disagreeable odor. Then finally, gases. Ethylene gas is a high penetrating gas which exudes sweet ethereal smell. It is used as a disinfectant, present and potential toxicity to human including mutagenicity and carcinogenicity. It is effective against all types of microbes including viruses and spores. Formaldehyde gas is widely used for fumigating operation theaters and other rooms. Beta propiolactone is a more effective method of fumigating purpose than formaldehyde. Thank you.